Hello everyone, my name is Ian McLaughlin and I have a PhD in neuroscience, and today I want to talk about another really impressive innovation in brain-computer interfaces. And so I've talked about brain-computer interfaces several times now, everything from the predictably famous Neuralink, started by Elon Musk, which is a bit of a drama-laden story, by the way, if you're interested in checking that out, all the way to a device called BrainGate, which has demonstrated perhaps more like tangible progress. And I have links to both of those down in the description below if you're interested in that progress that other groups beyond the one we're going to talk about today have made. Now, both of those companies have some pretty impressive and fun demonstrations of their technology, and really are worth checking out, but neither of them were focused on what this latest group has demonstrated, helping people who are blind to see again. So in short, Neuralink, at least thus far, has demonstrated an incredibly impressive miniaturized device, which you can see right there, that can be implanted and wirelessly listen and report the activities of neurons in a part of the brain that's associated with, at least according to their demonstration, with how a pig uses their snout to sort of like probe their environment, poking and sort of snuffling around their environment while they're exploring it. BrainGate, earlier this year, which is a collaboration between scientists and physicians at Brown and Stanford universities, as well as Massachusetts General Hospital, some of the top biomedical institutions in the world. So BrainGate was able to both wirelessly record the activity of neurons, but could also deliver stimulation. And, all wirelessly. Really impressive stuff. However, neither Neuralink nor BrainGate had entered the realm of visual perception, easily one of the most, if not the most, complicated component of our entire sensorium, right? It's kind of silly to try and rank like how complicated various aspects of our nervous system are, right? But I think it's pretty uncontroversial to suggest that vision is certainly the system that at least dominates our sensorium, right? We rely far more on vision than smell or taste or touch, or even our ability to hear, to get a sense of just what's happening around us. And of course that totally makes sense, right? It's the only sensory system that's updated by changes in our environment that literally happen at the speed of light, right? Our olfactory system our ability to smell operates at the speed of how long it takes molecules to spread through the air around us, which is pretty slow. And our auditory system, our ability to hear, is updated quite a bit more rapidly than that. At the speed of sound, right? The speed at which waves of air molecule, compressions propagate through the gas around us, hit our earballs, and then vibrate tiny little hair cells inside our head, right? Faster than the olfactory system for sure, but it's certainly quite a bit slower than the speed of light, because pretty much everything is, right? And so obviously our nervous system, doesn't operate at the speed of light. Far from it. In fact, and this has been tested and quantified, okay? So believe it or not. So, to be a bit clearer on this, while the photons that splash up against, our various stuff around us, right, in our environment, they're then sprayed out at us from the various devices and screens, right, that we use. While those photons are traveling at the speed of light, our nervous system operates at the speed of biology, right, which, to be clear, is quite a bit slower than the speed of light. And so there's a fun example that the neuroscientist David Eagleman uses in his book Incognito, which is I have right here. Just a fun book to read. Definitely recommend it to anyone who's just generally interested in kind of light, not too technical, or jargony laden exploration of some neuroscience. I recommend it. But anyways, he has a fun example of the relationship between the speed of photons and our nervous system. And so it takes about a half second for our conscious awareness to kind of grok that something has changed in our environment. But we humans are capable of responding to certain changes in our environment more rapidly than this time would presumably allow. And so one of the fun examples in Eagleman's book is hitting a baseball, a particularly fast baseball. A fastball, if you will. So Eagleman, at least in my opinion, is a lovely writer, but I won't just recite his whole example, which is on page 8 and 9, so right in the beginning. But he highlights that there was a fastball thrown by Nolan Ryan, and which was clocked at just under 101 miles per hour. And so as a result, that baseball crossed home plate in 0.4 of a second, right? It's just physics. And so 0.4 of a second is pretty fast. Faster, in fact, than our conscious awareness is actually capable of operating. And so, as Eagleman writes, This gives just enough time for light signals from the baseball to hit the batter's eye, work through the circuitry of the retina, activate successions of cells along the loopy superhighways of our visual system at the back of the head, cross the vast territories to the motor areas, and modify the contraction of muscles swinging the bat. And then Eagleman gets to the main point of his example. So, quote, amazingly, this entire sequence is possible in less than 0.4 of a second. Otherwise, no one would ever hit a fastball. But the surprising part is that conscious awareness takes longer than that, about a half second. So in other words, any batter who's capable of hitting a fastball at that speed, the speed that ball was reaching, using components of their nervous system, beyond just their visual system, right? That's the only way that they could do it. If they were only using their visual system and, keeping their eye on the ball, as my dad's sagely advice was when I played baseball, and he was absolutely right, of course, but if batters confronting pitches at these speeds were relying, solely on their eyes, it'd be physically impossible for them to actually react rapidly enough to swing before the ball was already in the catcher's glove. So how is that possible? So, it's really a topic for another day, but the point is that we clearly have a whole broad repertoire of systems that enable us to both detect and physically respond to events in our environment that don't require conscious awareness. So our conscious, unconscious processes are capable of some pretty impressive feats. It's a beautiful example. I couldn't help myself, but, in my humble opinion. And that's something that Eagleman is gifted at creating. But in case you're wondering, the fastest pitch of all time reached just over 105 miles per hour, about 169 kilometers per hour. Pretty bananas. By the way, I actually met David Eagleman several times when I was at Baylor College of Medicine in Houston while he was a professor there. Really nice guy. I actually just started participating in an effort that he'd started exploring, a kind of still emerging discipline called neurolaw, which basically confronts the increasing role that science, and particularly the science of consciousness in the brain, neuroscience, is playing in the legal system, particularly criminal law. And I'd gone to 2 meetings that he had at his lab, and I was getting pretty psyched about the whole project, just trying to figure out how I could, plug into his whole effort as a, basically the only cell molecular circuits person in the room, full of like cognitive neuroscience people and computer science people and lawyers. But then I ended up heading here to Philly. And as evidence, this copy of Incognito happens to be signed. Can you see that? Yeah, you can sort of see it. There we go. Anyways, definitely recommend this book to anybody who is interested in sort of like fun explorations of neuroscience. All right, so that whole jaunt down memory lane was at least intended to illustrate 2 things: how incredibly complex our visual system is, right, as well as how incredibly integrated and adaptive our broader sensory system and consciousness is, right, enabling us to respond to changes in our environment using vision, along with a whole repertoire of other capabilities to respond to those changes even faster than our visual system can allow for, according to the laws of physics, right? So a question that I've always had, before I got into neuroscience and everything, that I had, in high school, let's say, or whatever, even younger, it's always been, what is it like to be blind? Do folks without typical vision just experience complete darkness, right? And the reason that I wondered that was based on what I'd experienced, when I was in a very dark room with my eyes closed, even deprived of any source of visual stimulation, I could still sort of see things, right? I could, and I still can see, little flickers of what seemed like light, with my eyes closed and the lights off, and blackout curtains and everything, right? And it isn't like, someone pointing a flashlight at me with my eyes closed, right? The light has this whole variety of colors, and it occurs in what seem to be like geometric patterns, random geometric patterns, but patterns nonetheless, an interesting experience, right? And I bet you've experienced that at some point too, right? So that visual experience, in the absence of any actual photons splashing up against your eyes, is called a phosphene, okay? And so it's the perception of light in the absence of any light. And it's absolutely normal and natural. It's the byproduct of neurons in the part of the brain that processes vision becoming spontaneously active, starting to just randomly fire impulses, or sort of randomly. And so this, of course, presumes that the visual cortex, and we'll talk about where the visual cortex is in just a sec, but this presumes that the neurons in the visual cortex are still intact. They're still there, they're still healthy, they're still functioning. But the point is that even for folks who qualify as completely blind, right, those neurons are still there, but they're just not receiving that eyeball-based stimulation. And so it turns out that, and there are absolutely many different causes of blindness, and that blindness can manifest in multiple ways, to be clear, but it turns out that at least some people who are blind do not experience pure darkness. So rather, there's a perception of, quote, bright, colorful, ever-changing, often terribly distracting light. So that's a quote from an article written by someone who's designated as having no light perception, totally blind. And I've linked to his article down below. It's very interesting. Recommend reading it if you've ever wondered what I did, right? And it's interesting beyond his description of purely what his visual perception is like. So just definitely recommend checking it out. And that's what makes the achievement that this group that we're going to talk about today so impressive. So this is work coming out of Elche, Spain, which, by the way, seems like one of those outrageously beautiful parts of the world where people get to do like actual science in actual paradise. Like there are solid neuroscience labs in Nice in France, or I imagine UC San Diego is another place that's just, I couldn't imagine getting any work done, right? When the outdoors is just like perpetually nice and beautiful. But anyway, so that's where this group is. In Elche, at Miguel Hernandez University, specifically, in Paradise. And so there's a wonderful article in MIT Tech Review, to which I've also linked below, which explores this story. But before we get too into it, I think it's worth noting at the outset that there have been a lot of sort of exaggerated headlines about this particular innovation, like implying that they've cured blindness, and that's absolutely not what this is. But it's still really interesting and impressive. So fundamentally, what this group has done is to show that we can implant a brain-computer interface, which basically has a bunch of electrodes, little things that can stimulate neurons, record from neurons, making them fire, right? That they can do that. They can implant these electrodes right into part of the brain that enables us to understand what our eyes are seeing. And most interestingly, they did it with a human participant. And this is that generous human, Bernadetta Gomez. And so that's her wearing the sort of preliminary, beta tested, device, which basically just looks like some futuristic glasses, right? And they obviously have a bit of extra equipment going on, just beyond just some lenses. And so here's what the device, really is, a sort of schematic of it. And so the glasses have a camera right in the middle, right in the middle of the bridge of the glasses. And then so they have that fancy camera that then sends information to a device that processes the images through some filters that processes, video, right? And all of that ultimately ends up at their electrode array, which looks like this. So keep in mind, if you can see that little black bar on the bottom, that's 2 millimeters. So this is pretty small, right? I mean, that's obviously not 2 millimeters, but 2 millimeters is really, really quite small. And so they call this electrode array Serastim 96. And so astute listeners probably noticed that 96 number there. And so believe it or not, that's not just some random number that they're including to make the device sound particularly advanced. It's not like the Vision Generator 5000 or whatever. The 96 refers to the number of electrodes that's on the electrode array. Okay, and so here, wait, where am I? Boom, boom, boom. And so, interestingly, while they theoretically had all of those electrodes at their disposal, right? Oops, there. They had 96 electrodes at their disposal. They could only stimulate with 16 of them at a time, right, to stimulate neurons. And they were stimulating neurons in a part of the brain that processes visual perception. And so, by the way, the part of the brain that does that processes visual information, is rather inconveniently distal from our eyebrows, or eyeballs. Eyebrows too, I suppose. But so we have all these photons, right, splashing up against our eyeballs, and all of this neurophysiology has to not only process all of those patterns of ever-changing, photons, changing at the speed of light, patterns that are, that completely unpredictable. There's no way that the eyes can actually just foresee what they're seeing, right? But so these patterns are changing at the speed of light, splashing up against our eyeballs. But then all of that information has to be translated to exactly the opposite side of our brain, right? Sort of a silly way to design it, huh? And so anyways, this is their device, right? And so they were working with Bernadetta, who had been blind for 16 years, over 16 years. And she's 57 years old. She lost her vision at 42 due to basically damage to the neurophysiology that connects the eyes to the visual cortex, right? They have all that distance to traverse. And so she progressively lost her vision. And so, she was born with vision, right? But then she lost it. And it was lost to the extent that she was completely incapable of detecting any light whatsoever. And so Bernadetta had this electrode array temporarily implanted in her occipital lobe right there. right at the back of the brain, and was capable of seeing light for a 6-month period after the implant. And so I'm going to quote from that wonderful article in the MIT Tech Review, quote, using the implant, Gomez identified ceiling lights, letters, basic shapes printed on paper, and people. She even played a simple Pac-Man-like game piped directly into her brain. 4 days a week for the duration of the experiment, Gomez was led to a lab by her sighted husband and hooked into the system. End quote. So how cool is that? Particularly the Pac-Man thing. That's pretty wild, directly into your brain. And so there's another quote from the article that illustrates how counterintuitively casual the implantation process is. So Gomez is sitting in a room with the scientist who devised the device, right, after the completion of her experiment. And there's another person who's blind in the room, and they're evaluating whether they'd like to participate in the way that Bernadetta did. And so this is the quote. At one point during this meeting, as Fernandez, the lead scientist, explains how the hardware connects to the skull, Gomez interrupts the discussion, tilts forward, places the prospect's hand on the back of her head where a metal outlet used to be. Today, there's virtually no evidence of the port. The implant surgery was so uneventful, she says, that she came to the lab the very next day to get plugged in and start the experiments. She had no problems or pain since. End quote. That's pretty awesome, right? I mean, I'd absolutely expect that there'd be at least a bit of a recovery process, like ice cream and comfort movies for at least a week. I demand that, right? But anyway, so this group based the way that they'd stimulate, the occipital lobe or the visual cortex on an interesting strategy. So they were learning how the human eye sends signals to the visual cortex, right? By literally measuring signals from the eyes of recently deceased organ donors. And so because it turns out the human retina can only be kept alive for about 7 hours. So there's a lot of like late night calls to the lab where they had to go and get those retinae. And so, yeah, and it can only, the retina can only survive for about 7 hours outside of your head. But so anyway, so that's how they figured out how to stimulate the visual cortex, what patterns to use, how far apart the electrodes would have to be, that kind of stuff. And so she was able to decipher letters and basic shapes with this implant, as you can see in this figure from their paper here. And so up top, right there under A, you're literally seeing, which of the electrodes, which of the 96 electrodes we're sending activation, we're stimulating neurons, close to them in Bernadetta's visual cortex. And so you're seeing the difference between like what a capital O looks like versus a lowercase o. Pretty similar shapes, but slightly different, right? And as you can see over time on the bottom there, Bernadetta grew to get better and better at discriminating just that subtle difference, reaching 100% accuracy after just a week. It's pretty awesome. And so they also did a kind of real-time test of how effectively Bernadetta was able to discriminate between stimulation on the right or the left of the implant. And this is pretty fun. But so first of all, this is what it looked like. This is a video. So this is, an example of what one of their experiments looked like. That's Bernadetta. So fundamentally, you're seeing Bernadetta sitting down with her eyes closed, right? Doesn't necessarily matter that they're closed, but that's how she kept them. And, and she's plugged into the system, right? Up top, you can see that here. And so you're seeing a black box up top with Maggie Simpson from the TV show The Simpsons periodically appearing and firing a shot at you. And she's either using her right or her left hand. And so depending on which hand she's using, the implanted electrodes will stimulate certain neurons to correlate with right or left. And you can see that in the middle. That's the electrode array. And so then, as soon as she could, Bernadetta presses a button for either the right or the left to get a sense of whether the implant was able to successfully and consistently experience that sort of deliver that experience of that the visual perception on either the right or the left hand side, right? And by the way, if I'm reading this study correctly, it's not like she was seeing Maggie Simpson. Maggie is just for our benefit, I believe, but I'm not 100% sure on that, pretty sure. But that's basically, enough to perceive things like letters, sidewalks, hallways, just very general visual experiences. And that can be super helpful, right? However, keep in mind that this was just one teeny tiny little implant. Just, one of them. And so, imagine implanting significantly more. That's a sort of Wild West, right? Because it's hard to know at what point the artificial stimulation departs so significantly from our natural visual system that it starts to just seem like hyped up phosphenes, right? You're just cranking up the volume, the gain so high that you begin to experience TV snow, as Russ Jaskalian describes it in that MIT Tech Review article. And so Fernandez, the lead scientist, makes the perfect analogy. So this device is to vision as a cochlear implant is to hearing, right? Cochlear implants are essentially microphones, that function essentially just like the microphone I'm using right now, that are wired directly into the inner ear, bypassing part of the auditory system that normally enables us to hear things, directly stimulating the part of the brain that detects sound. Right? Well, that's essentially exactly what's going on here. Evidently, Bernadetta would have liked to have kept the implant longer than the experiment permitted. And she's also planning on framing and hanging her implant once the team of scientists is done studying it. And I'm sure they're studying it just to get a sense of how much the materials degraded over that 6-month period, as well as perhaps any footprints from Bernadetta's own immune system, right? That might have been left on the implant, right? The longer there's some foreign body that's in our body anywhere, but essentially, the more time the immune system has to just attack it, right? It's not really attacking it, it's just inevitably going to recognize foreign, right? Or not me. And then, deploy whatever innate immune system activity it can, right? So, and that's predictable, that's known, they will anticipate that happening. So I'm sure that they're evaluating to what degree and we can broadly call that inflammatory, right? So I'm sure that they're studying to what degree this tiny little implant was inflammatory and to what degree it degraded over time. And so, but given this group's success to date, it turns out that they already have plans to work with 5 more people who are blind. And so remember, that article was written by someone who, so you remember that article that I referenced in the beginning, written by somebody who has, blindness that's designated as no light perception, right? Completely blind. Well, part of the reason that his article is so insightful is that, he gets into the reasons that blindness is debilitating beyond purely the inability to see. And so, as I noted, right, he describes the constant perception of spontaneous geometric patterns of light despite being blind. Constant perception of light. Think about that. It's constant, meaning it's inescapable. So he can't close his eyes to blot out the perception, right? It's just always there, constantly. Can you imagine something like that? Like just try to imagine never being able to, stop seeing something. Try to imagine any sensory experience that you just couldn't stop, right? Imagine an odor that you could not stop smelling, or even just a source of pressure somewhere on your body, right? Just pushing on you constantly that you could not stop feeling, right? Or a sound that you couldn't stop hearing, like tinnitus, right? Which is quite terrible and can be debilitating for some folks. I don't know about you, but before I'd really explored accounts from people like Damon Rose, who wrote that article about what it's like to be blind, I hadn't really contemplated the sort of secondary and tertiary ramifications of losing vision. I hadn't really thought about the challenges that they face beyond just having to adapt to not being able to see their surroundings. And so that's why I find this research so compelling. Right? Not only is it helping individuals who are living without access to a part of the universe that's, quite literally delivers the color of our surroundings, but it's also some of the early steps towards helping to anchor their sensorium, right? Such that their visual system isn't just a byproduct of the essentially random whims of neuronal activation generating those phosphenes. And then, of course, also it is meaningful progress in the direction of some pretty sophisticated brain-computer interfaces, the applications of which pretty wild. All right, that's what I have for you today. If you dig this kind of discussion of neuroscience and biotechnology, maybe you'll consider subscribing and liking and pressing all those helpful internet buttons. Otherwise, have a great week. I'll be here next week with another interesting instance of progress in biomedical science. Thanks for joining. Oh, watchmaker, I didn't understand what she saw. Was it the flash from a gun? The movement. Okay, yeah. Here, we'll bring it back up. Oh, that's the wrong one. Okay, so yeah, it is a little odd the way they decided to demonstrate this. Okay, so up here you're seeing a cartoon of Maggie Simpson, right? In the middle you're seeing the actual electrodes that are being stimulated. So keep in mind, right, she can't see really complicated I think the sort of comparison was, it's almost like 10 by 10 pixels, right? So not exactly, this is 1280 by 720, this video right now, so it's a lot more. And so, those are the pixels. So essentially, you can see the pixels. They correspond to specific electrodes, but then and each electrode is basically able to stimulate about 1 to 4 neurons, which is great. And then on the bottom, you're just seeing her pressing the buttons. And so what she is seeing is just a flash of light, essentially. And it, it doesn't look like a bullet, doesn't look like a gun or anything like that. It looks like probably a fair bit like somebody pointing a flashlight in your face, but more sort of limited, right? Or maybe even more like it wouldn't actually look like this, but it's sort of like a laser pointer, right? Where it's like this very defined region of stimulation. That's almost certainly what she's seeing, and it's just like on top of everything, right? So yeah, so it's really more of like an indicator, like something's happening, or I detect a visual experience. That's really it. But as I said, right, so this is just 96 channels, 96 electrodes, 10 by 10 essentially pixels. You can, and keep in mind, these are super, super small, so you could do 5 of them, 10 of them, 500 of them, who knows. And they're not that invasive. Like when we look at the actual device, they're not that invasive. It looks a little bit more menacing than it is. Like if you were to put that on your finger, you probably wouldn't have any idea that there's actually little electrodes. You'd probably just feel like putting a dye on your finger or something like that, but much smaller. And so, they just barely penetrate into the brain. It's not like electrodes, going deep. So, pretty negligible, frankly, as far as brain surgery goes. But yeah, so that's basically the experience. Thanks for prompting me to clarify. I'm sure you were not alone on that. All right, yeah, so thanks for watching, and hopefully I'll see you all next week.